Good morning, student. Today we will discuss about the digestive system of mammals. In previous lecture we discussed about the digestive system. Today we learn about the digestion process. Digestion is the process of mechanical and enzymatically breaking down food into substance for absorption into the bloodstream and transport to the body organs. Digestion is mainly often broken down into two types. First type is mechanical digestion. First type is mechanical digestion. In this type, food is physically broken into smaller parts. Example, chewing. Second type digestion is chemical digestion. Chemical digestion. In this process, food is broken down by acids and enzymes into the basic units. This digestion starts from the mouth. Digestion begins even before the food enters the mouth. The smell or even thought of food starts the production of saliva by the salivary gland. Once the food is inside the mouth, it is moistened by saliva and the teeth and tongue begins the process of mechanical digestion. Saliva contains an enzyme called as salivary amylase. Saliva contains enzyme called as salivary amylase, which breaks starch. Saliva also contains mucus that helps ease the passage of food through the digestive system. Saliva is an important digestive agent and it contains two digestive enzymes. These are ptyalin and maltase. The salivary amylase contains two digestive enzymes, that is ptyalin and maltase. Ptyalin and maltase. These ptyalin act on starch. Ptyalin act on starch and convert it into maltose. On maltose, these maltase enzyme act and these converted into glucose. Maltase enzyme act on maltose and convert it into glucose. Once mastication and amylase digestion are complete, the food will have become a small round structure which is known as bolus. This round structure known as bolus. Peristaltic waves is the slow contraction of smooth muscles around the pipe of digestive system. Slow waves of contraction runs along the gut, pushing the bolus along the right direction away from the mouth towards the anus. Digestion in stomach. After the food has been swallowed, it passes through the esophagus to the stomach, where it undergoes further mechanical and chemical changes, and these digestion of stomach, it is called as gastric digestion. The bolus enter into the stomach through the muscular wall at the top called as cardiac sphincter, and this sphincter controls how much food enters in the stomach and when chemical digestion are due to gastric juice. Gastric juice are the secretion of gastric glands, and gastric juice contains mostly HCl. Gastric juice contains mainly HCl. These gastric juice, the wall of stomach has numerous deeply pitted gastric glands which secrete gastric juice, and these about 2 to 3 liters of gastric juice is secreted daily by the normal adult. Normal adult mainly secretes 2 to 3 liters gastric juice, and this gastric juice mainly contains HCl, and these HCl secretes parietal cells of stomach wall, and their concentration is about 0.16 normal concentration of HCl secreted. These HCl mainly function as these dissolve the living material between the cells in order to make readily available for enzyme action. HCl it also acts as a bactericide, and harmful bacteria are killed, and HCl provides acidic media for digestion of the food material. In HCl gastric juice, again inactive pepsinogen is present. This HCl is acid that is strong enough to dissolve razor blades, and pepsin an enzyme that break down into proteins. Gastric juice mainly contains 2 components, HCl and second part is pepsin, and this pepsin is proteolytic enzyme. These act on protein and protein breakdown takes place. Both these chemicals could potentially harm the lining of stomach, so it produces a slimy mucus layer to protect itself from the damage. In this gastric juice, here pepsinogen is inactive form. Pepsinogen is inactive form. In presence of HCl, pepsinogen converted into active pepsin. Pepsinogen converted into active pepsin, and this active pepsin acts on protein, and this active pepsin acts on proteins, and proteins are converted into peptones. Proteins are converted into peptones. This gastric juice also contains renin, and this renin present in the cup, and this renin also in the form of inactive renin. It is called as prorenin. Prorenin in presence of HCl, the inactive prorenin converted into renin, converted into active renin, and this active renin, again this active renin reacts with protein casein. This active renin combines with protein casein from milk protein, and these are converted into the paracasein. This renin act on milk protein and converted into paracasein. Paracasein react with calcium and converted into calcium paracasinate. Converted into calcium paracasinate. In this gastric juice, again gastric lipase is present, and this gastric lipase acts on the neutral fats and converted into molecules of glycerol and 3 molecules of fatty acids. In stomach, peristaltic waves continues, which helps to mix the food with the gastric juice. Not many compounds are absorbed into the blood from the stomach. After 1 to 2 hours in the stomach, the food is thick paste referred to as cham. After 1 to 2 hours, the food is converted into a pasty form. It is called as cham, and this cham pass towards the small intestine. It leaves the stomach through the pyloric sphincter at the bottom of stomach, and the cham transported towards the small intestine. This is all about the digestion in the stomach. Thank you.